All right, we are now recording. Oh, now the important stuff. All right. So we're about to start talking more about evolution, but now how we would look at it in a population setting, the evidence we would use, the actual population genetics, things of that nature. So kind of the math or the real science behind what we're going to talk about. All right. So if I were to ask you the question, can an individual evolve, what would you tell me? Could be a trick question, right? Could an individual evolve? Lock you out in 5, 4, 3, 2, 1. Time. So, do individuals evolve or do populations evolve? That's the real question, right? Populations, okay? So it wasn't really a trick question. So individuals cannot evolve, but I made you think it was a trick question, right? It was a double negative trick. It wasn't really negative though, was it? Yeah. So a population is all the members of a single species occupying a particular area at the same time. So we are indeed a population in this room. We are all human beings, most of us, right? Ask Joe, he says I'm a vampire. I don't know how that works, but I'm going to go with it. Diversity exists among members of a population, and population genetics is the study of this diversity in terms of allele frequencies and differences, and evaluations of the diversity of a population by studying genotype and phenotype frequencies over time. So here's another question for you. Does allele frequency change? Does allele frequency change? Boy, that's a loaded question. Does allele frequency change? Let me restate that. Does allele frequency usually change? No, I might just be messing with you, too. Pretty good at that. Go with your gut, right? Go with your gut. In 5, 4, 3, 2, 1. Ooh, the perfect 8. Oh, that's lucky or something. I don't know. It's not, though. It's 8. Crazy 8s. All right, so the best answer is not usually. Usually the alleles in a population remain relatively stable. It's just we juggle them around, right? But the alleles are always there. You don't really get rid of alleles very often in a fairly stable environment. Most environments are stable for the most part. Okay, we're going to talk more about that. So in the 1930s, population geneticists began to describe variations in a population in terms of alleles. And if I ask you what an allele is, what would you tell me? What is an allele? Keep you busy. Make you think. What is an allele? Anybody else hang up Halloween lights this weekend? Oh, my word. I agree. I have to use the ladder and just keep up, appearances. You can't have.
Halloween, you gotta take advantage of it.
Yep. I do have these dead birds on the wire. Did I tell you guys about the dead birds on the wire?
Oh yeah.
Oh my word. There's one left. He's just hanging. Oh, he looks like a bat. I mean, it was a bird, but because he's all like bones now. He's just all shriveled up and hanging there. I've never seen anything like that. What? Well, it is creepy. Like this. No. Gosh, no.
That's just what a vampire would say.
See, I can't even argue this. So a gene thingy, an alternate form of perfect, a different form of a gene, dominant recessive genes, they can be, form of a gene, a version of a gene, right? Okay. Part of a chromosome, a gene. Did you guys hear about the new DNA that came out the other day? The new DNA that came out the other day? It's a designer gene. Oh, it's bad. So then we look at microevolution as it pertains to evolutionary changes within a population. Microevolution, that is not changing from one species into another. That's the small varying versions of alleles or genes within the same population. So various alleles at all the gene loci or location in all individuals make up the gene pool of the population. So if I counted up all the different alleles in a population, that would be my gene pool. I just call it the laundry, right? Throw all your genes in the washer. It's like a gene pool. So the gene pool of a population can be described in terms of genotype frequencies or allele frequencies. We are going to be doing what's called the Hardy-Weinberg principle in here quite a bit. We'll be doing a POGIL over that, just one, I promise. Okay. But it might be one of the more important ones we do all year, because I always see that on the AP exams and on the test exams. If I add up, here's a new question for you. If I add up every single one of the alleles, percentage-wise in a population, what will they add up to? So easy, it's hard. If I were to add all of the population, alleles in a population, what would they add up to? 100% or 1. Very good. I didn't waste too much time on that one. Now, that seems like a very simple thing, but it is the rule of the Hardy-Weinberg, and it's like our known. Like, yes, everything equals 100% or 1. And if I know part of 1, I could probably use a little bit of simple algebra to figure out the rest, right? And that's what the Hardy-Weinberg principle states in kind of layman's terms. So what that states is when I have phenotypes, I don't always know the genotype behind them, right? So here we have a moth. You guys probably remember the peppered moth experiment. But the homozygous dominant and the heterozygous, both are going to show the same phenotype, but I will have a different genotype underneath. Now, if I do have homozygosity, I am able to measure allele recessiveness, right? How many alleles are present in all of these right here? 2, 4, 6, right? Okay? And so now when I start to think about allele frequency, I start to think about it a little differently. It's not just counting the phenotypes that I see. I have to take into account all of the different combinations mathematically for homozygous dominant, heterozygosity, and homozygous recessive. And this leads into the frequency of alleles, how often they show up. So the proportion of each allele within a population gene pool, where the frequency of the dominant and recessive allele must always add up to 1. And this relationship is described by the expression P plus Q equals 1. P referring to one allele, Q referring to the other. So microevolution involves a change in these allele frequencies. Normally, do you get a shift in allele frequency? You guys all told me kind of half and half, right? Normally, no. But over a long period of time, in changing conditions, yes. Now, if the gene frequencies do not change over time, microevolution has not occurred. Which means if I come back 100 years from now and the human population looks exactly the same way, like I have the same number of Native Americans, the same number of Irish people, the same number of Asians, the same number of Africans, if I have the same number of all these subspecies, then we say microevolution has not occurred. Has microevolution occurred in the human population in the last 100 years? Heck yeah. Okay? Especially in America. We are the melting pot, right? Think about it. The whole thing with like the Industrial Revolution, people starting to move all over the globe was pretty big in that. So when I look at the Hardy-Weinberg, I'm counting all of the different phenotypes first. And that gives me some of the knowns, but it doesn't give me all of them. I need to blow this up a little bit. I know I do. So we don't always know heterozygosity, but we always know this homozygosity, and we'll always be able to count them out and get actual numbers for that particular equation. And this equation will come into play when we do the POGIL today. But if P plus Q equals 1, then we can also say, in relationship to our alleles, that P squared times 2PQ, because that's our heterozygosity, times Q squared equals 1. Okay? And what that'll do is it will allow us to apply that P plus Q equaling 1, looking at just dominant and recessive alleles, to a situation where we do have heterozygosity. Okay? And then we can find out the total allele frequency for both dominant and recessive alleles. Does that make sense? It's just saying that if P plus Q equals 1, looking at a dominant and a recessive allele, if I apply the same idea, the same fact really, that all of that equals 1, and I know that I have a situation where some individuals are going to be heterozygous, I know that P squared plus 2PQ plus Q squared is going to also equal 1. And knowing that is going to allow me to go back and do some basic algebra, because I can figure out Q squared, right? Because Q squared is who? Recessive. Okay. And if I can figure out Q squared, I can also find out the Q for 2PQ, right? And we'll do this together, don't worry, which will allow me to figure out P here and come back and find out P squared, if that makes sense. I did, yes. And I zoomed in and I shouldn't have. Now I'm zoomed in and I can't zoom out. So the Hardy-Weinberg statement, or the Hardy-Weinberg equilibrium, says that allele frequency in a population will remain constant, assuming no mutations happen, no gene flow occurs, random mating does not exist, does exist, no genetic drift happens, and no selection occurs. So is the Hardy-Weinberg equation ever really going to happen over time? The answer there is no. But can I take a snapshot of a population and see if genetic drift is occurring by viewing allele frequency? I can. And so if you use the Hardy-Weinberg equation to measure allele frequency and then to see if there's changes in it, you can then measure evolution, microevolution. Does that make sense? Okay. Well, sure is a terrible answer. Does that make sense, yes or no? I'll ask. I hate Sures. Sure is the deodorant that might not work. Does that make sense to you? If it doesn't, I need to re-explain it in a different way. The idea that if allele frequencies are changing, I can then have a tool to measure microevolution. Those small changes and subtle differences. So I have some no's. Let me kind of give you an example of that. If I have the human population in this room and we just look at eye color, we can measure that, right, in a phenotype. And if I can measure the number of us that have blue eyes, I know how to measure a recessive trait in this room. I now know Q squared. I also know there's brown-eyed people in the room, and if this is an interbreeding population, I'm going to have homozygous dominant and heterozygosity here as well. Because of that, I can find out what the heterozygous portion of that Q is to inversely find out what the P is. And we're going to do this. Don't worry. This is not something we're learning just yet. We're going to do that with a POGIL and go over it quite detailed. And so by doing that, I can then find out how many dominant alleles are in this room, referring to just brown and blue eyes. Now, I know there's other alleles with eye color, and that's not a trait that's just influenced by one gene. But pretending it were, I would be able to measure next year, if we were a completely different population, if the frequency was changing. Those would be small changes in my entire population, and if the allele frequency changed, I would know I'm experiencing microevolution. Here's the question, and this is the thing that really you need to take home from this. Can I measure evolution by just looking at the phenotypes in a population? I'll ask, just go ahead and just pretend I asked it. Can I measure microevolution by just looking at the phenotypes in a population? And you can't. Because the allele frequency is the true telltale sign. And you can never just tell it by looking at a population because of the dominant recessive thing. Okay. Like you would never know that I carry a blue-eye gene, but I do. So when we talk about the Hardy-Weinberg principle, we're basically looking at sperm and egg and the allele combination of the two. And remember, there's that basic chance of receiving part of a sperm from dad or part of an egg from mom. And if dad is heterozygous and mom is heterozygous, we know we have the 1 to 2 to 1 genotypic ratio variability. So based on this idea, we can add ratios, and these are the ratios that we're looking for when we do our population counts for our Q squared. Okay? So if I know this guy right here, can I turn around and find out the rest for everything else? You can, right? Because if I can find out Q, I can find out what portion of Q 2pq is, and then I can flip around and find out the P of the 2pq and also figure out P squared. Okay? And we're going to do that, don't worry. So the Hardy-Weinberg equilibrium, it's where you have required conditions that are rarely, if ever, met. So sometimes on islands you see this a lot more because it's a very stable, not really changing environment usually. But deviations from a Hardy-Weinberg equilibrium indicate that evolution has taken place. Meaning if I see a shift in allele frequency, I can measure evolution at a small scale. And remember, I'm not talking about macroevolution yet. We're talking about small changes, subspecies of dogs, subspecies of humans, that kind of thing. And an analysis of allele changes in populations over time determines the extent to which evolution has occurred. So we can measure this over long periods of time. We could take this measurement every year for over like a 100-year period, and I would be able to actually prove that microevolution occurs. We do this in a lot of different ways. We do it with humans. So looking at these things that cause the Hardy-Weinberg principle to hold true, you would have to isolate all of these variables. We know it's impossible to. But these are also the driving forces behind evolution. So we know that having non-random mating would allow complete variability and complete autonomy to the whole process of mixing up genes. Now, there are very few species that do random mating. But the fact that we have non-random mating as humans does drive a shift in allele frequencies. Does that make sense? If I'm choosing for a trait, I'm now choosing a phenotype, I'm now choosing alleles. And by doing that, you're not allowing the Hardy-Weinberg principle to hold true. So in this case, you are going to get a change in the genotype frequencies. Now, if I look at no selection, meaning the environment does not do any selecting, well, we know that doesn't happen either. But it would be in a perfectly stable environment where certain alleles are selected for and against. In this, you would get a change in allele frequencies. And so this would drive evolution if selection did occur. Whereas there's no mutation, we know mutations happen naturally. It is the driving force behind evolution. This is where you add new alleles. Would adding a new allele to a population change its frequency? Duh. Yeah, right. It's a new one. Okay. So that would definitely drive evolution. It is the driving force behind evolution. No migration, that's pretty rare, but in an island situation, thinking about our Galapagos finches, that is what the driving force of that evolution was, where it isolated them. So individuals can carry alleles into or out of the population. So if I do migrate into another population, that can cause a shift of alleles. And then if I look at large populations where no genetic drift occurs, I say large populations because if I have a very large gene pool, I'm less apt to get a shift in it. Whereas if something changes in a small population, it can drive the whole allele frequency in one direction. Okay? It's almost like having a bunch of extra things just in case, like in a large population for biodiversity. All right. I know so much of this seems like a little bit of common sense, but unless we clearly state it and link it to frequencies of allele shifting. So looking at the different mechanisms of microevolution, we talked about allele and genotype frequency. Okay. Now, I want to do a little review of generations because we're going to be talking about a few generations. The first generation, the P generation, is what? 2 pure generations, right? Okay, so 2 pure generations would look like this. Let's use the same alleles here. There's a pure and there's a recessive pure, right? Okay. So in this situation, these two are going to create an F1 generation where all children will be what? Heterozygous, right? Okay. If this is my P generation, all F1 generation, just remember from genetics, I'm doing this just to review real quick, your entire F1 generation is going to end up being heterozygous. I'll let this zoom in. I can't see very well here. So if I look at the allele frequencies here, between these two, and I say that the dominant is 0.20 and the recessive here is 0.80, both equaling 1, of course, we can come to that by looking at the actual genotype frequencies. And you have to look, yes?
So on that first pie chart, if 3 of the offspring are going to have the dominant allele, and why is that 0.2?
Where are we at? Right. Well, this is what we get from this. I got to go here first. Does that make sense? It's kind of working backwards. So if I come over here and you see how you end up getting the allele frequency based on the genotype frequency, that's what we need to do. And so that's kind of what we're going to be, well, it is what we're going to be doing in the lab. So if I start with the equation where you have p squared plus 2pq plus q squared equaling 1, and I know I have 0.64 as my homozygous recessive, I know part of the equation, so I can plug that in, right? Okay. And if I know my homozygosity is 0.32, I can plug that in as well. Okay. And the reason I know that is because I have a portion of that from 0.64, half of that, and then I end up with my homozygous dominant at 0.04, very rare, because that's what's left over once I run through the p squared plus 2pq plus pq squared. Now, what we're going to do, this is the same, should be the same equation over here. I don't like how this is not big enough to see. You guys can't see that very well, can you? Yeah. Right here? Portions of them will, yes. And that's what we're going to go through with the POGO. I know it's not good unless you do one with me, but and we're going to do that. But right here, you would be given certain phenotype frequencies. You would be given the phenotypes at least of the dominant and the recessive, and that is enough to figure everything else out, okay? So yes, you'll be given a lot of that information, okay? So assuming there's no change in the frequencies, we can see the Hardy-Weinberg principle hold true, but it's not always that easy. Oop, now I'm just drawing. Where'd my, oh, I'm still zoomed in, that's why. Sorry. Okay. So if we do see a shift from one generation to the next, and so we start to see a difference in our allele frequencies, in this case we went from 0.20 to 0.80, that's a huge allele frequency shift. But we would not want to take that from just looking at a phenotype. You'd want to actually find out the actual allele frequency of both dominant and recessive alleles, not just look at the phenotypic frequency. Now the causes of microevolution are kind of, they're not no-brainers, but in the way we've already talked about them before. Genetic mutations are huge. We know that in a small way, literally. The raw material for evolutionary change is a mutation. They provide new alleles, and some mutations might be more adaptive than others. Genetic mutations affecting pigment color in peppered moths have provided the variation needed for natural selection to occur. You guys are going to be doing a little bit of selection yourself by growing some bacteria. We're going to be culturing E. coli hopefully here in not too far off future. And then there's, of course, gene flow, where you move alleles between two different populations or more. And gametes or seeds in plants are carried into another population by the wind, water, other animals, defecation. Breeding individuals migrate into or out of a population. See that right now in our deer population. And continual gene flow reduces genetic divergence between populations. So I've never seen a wild animal stop moving, really. And they just keep moving around and migrating and mixing up their gene pool. Yep. This one? Okay. You got it? Okay. And don't worry too much about the Hardy-Weinberg. We're going to do a whole activity for that. Okay. You won't get that until you actually go through 1 or 2 or 5. Or 8. So then there's genetic drift, where changes in the allele frequency of a population due to changes rather than selection by the environment. And this does not necessarily lead to adaptation to the environment. And this occurs by disproportionate random sampling from the population. It can cause the gene pools of two isolated populations to become dissimilar. And some alleles are lost and others became fixed or unopposed. So we're kind of a lot like that as human evolution happened. We don't have a lot of others like us. We kind of just jumped into our own little really random species in a way. We did have other early hominids and things like that were similar to us, but nothing really close to us to really oppose us, I guess, if that makes sense. Like no one else had conscious thought, which is kind of unique to humans. And this almost likely occurs after what's called the bottleneck, where you drive all of the genes down into, think of an upside-down bottle. It's where you end up with one allele that's being chosen for over the others. When forced together, you get severe inbreeding. Those types of things happen. Now, when we think about it as in humans, it's just gross. But when you think about it as plants, it's no big deal, right? And that's the same situation. So when founders start a new population, this is usually what happens. Does inbreeding cause a lot of mutations and variations? Yes. Gross, yet part of what really does drive new versions. Think about the pug, right? That's mildly mutated, right? Okay. So stronger effect in small populations. The smaller your population is, the more extreme mutations you're going to get. Look at our dog. Our dogs are a great example of it, because look at all the variety you got out of forcing mutations and choosing the ones you wanted. It's really depressing when you think about what we did to the gray wolf, right? Maybe. Yeah. I mean, you go from the most ferocious predator to a Taco Bell dog, you know. So then the bottleneck effect, that's just referring to a random event that prevents a majority of individuals from entering the next generation. Could be caused by a meteor hitting the planet. Could be caused by pollution. It could be caused by deforestation. Could be caused by a lot of things. But the next generation is composed of alleles that just happen to make it. Do they have to be the strongest ones? No. They just have to survive and reproduce. I always talk about the two-toed sloth, right? Like, what are you doing here? You don't even move. He moves so slow, he grows like moss and colonies of lichen on him, and he carries around his own stinky ecosystem with him. But that's probably why he made it too, right? So, take a bite out of that, you're going to spit it out. Now, when a new population is started from just a few individuals, we call that the founder effect. That's an easy one to remember, like the founding fathers. Shouldn't make that comparison when we're talking about inbreeding. So the alleles carried by population founders are dictated by chance. And formerly rare alleles will either occur at a higher frequency in a new population or be absent in a new population. So often it's that rare allele, like let's say the really tall giraffe, that all of a sudden, you know, you can think about if you had a tall neck and you always had to reach farther to get your food off the ground or around, you probably were at the bottom of the totem pole, but then as vegetation shifted, right, all of a sudden you took a huge shift, and then all of a sudden your buddies were dying, you were eating lunch. You know, I mean, but that's essentially what happened, slowly but surely. Weird to think about, right? Because there did used to be short-necked giraffes. So again, that's just referring to the bottleneck. And bottleneck is referring, and if you think about that end, it's like a limiting thing that would have happened to create a new choice in alleles, so to speak, where it only let so many through. And sometimes it's those rare ones that survive, but a lot of times the rare ones get eliminated as well. Think about the really short end of the giraffe neck. You know, that one got eliminated in that process too. So, again, always looking at allele frequency, right? Never looking at the phenotypes of the population. So here you see that shift in alleles, so you know microevolution occurred. Okay? If you do see a shift in allele frequency, you know microevolution is happening. So then that leads into non-random mating. We talked about that, when individuals do not choose mates randomly. A good example of that's going on right now in my backyard is salmon. I said I got salmon coming up in my backyard right now, and they drop their eggs off, and then the men come by, and then they spray the eggs with their spawn. Is that random mating? I'll ask the question. Is that random mating? Yes or no? Oh, did I? Well, shoot, darn. Forget about it then. Oh, is that? Okay, then I should ask it. Is that random mating? Salmon spawning, random mating. Let's go through a few real quick. Give you some illustrative examples. Salmon spawning, is that random mating? One says no. Anybody want to be brave and tell me why it's not random mating? Who put no? What? Okay. You would say it is non-random or random? Completely random, right? Yeah, because, yeah. So it is random. You know, the female comes up, drops off, you know, 1 million eggs, whatever. Well, feeds 100,000 eggs. And then the male does spray them with millions of spawn. And they don't choose which egg finds which spawn or sperm, right? No. And then they die, right? And then, yeah, there's no thought in that, okay? How about this? 2 deer fighting for the herd, and then the one buck gets to mate all the does. Random or non-random? Is that an example? That is an example of random mating. I'll make that statement. That is an example of random mating. I believe the answer should be no, right? That is definitely not random mating. That's choosing, okay? There's competition involved there. The doe makes the choice to obviously go with the one that wins, that kind of thing. How about the birds of paradise? The birds of paradise are randomly mating. I'm going to make that statement. The birds of paradise are randomly mating. If you don't know what they are, they're the ones that dance for each other. Ever see them? Yeah. Yeah, they build a hut and then they dance in front of it and then they flash at each other. They're like teenagers at homecoming. It's ridiculous. Right? So the birds of paradise, that is again easy. Take it back 3 notches. I didn't even hear it and I know it was bad. All right.
I'm like Joe at homecoming.
Oh, okay.
Wow.
You okay? You want to talk to Miss Sissel about it?
I'm good.
No? Okay, all right. You tell me, you know.
I think you'd be the last person to know that.
Me?
Yeah.
I should handle the hotline, shouldn't I? I'd be good at it. I'd be fired a day into that game. You cannot do this anymore. What? I would. That's the problem. I would just, it's like, really? This is what we're worried about? Where are you going to college? I give them something to really worry about. Oh, that's terrible. And I'm recording myself. What? At any rate, individuals select mates with the same phenotypes with respect to a certain characteristic. Individuals reject mates with different phenotypes. We do that as humans, right? You know, no one says, I want to grow up and find me a couch potato, right? No one says that, okay? Increases the frequency of homozygotes for certain loci or location. So that means that we have more of a certain allele on a certain location of our chromosome. That's what that's referring to. Since AP biology, they're going to use that terminology, so we got to use it here. Locus, or like you think of locus of control or loci. All right, I'm going to pause there. I won't beat you up on a Monday.